All right, good morning, everybody. We have another new video. What? Two videos in 1 week? Must be just another manic Monday. All right, guys, so now we're going to talk a little bit about gel electrophoresis. So let's imagine we have a little small tube with some clear liquid in it. We know the liquid has DNA strands of a bunch of different lengths, but we need to sort them out by size. How are we going to do it? I don't know. Maybe we can use gel electrophoresis. So going back, guys, we already extracted our DNA. Remember, we did that in the previous lab. We extract it, we purified it, we got what we need. Now we need to separate it out by size. And we're going to use gel electrophoresis in order to do that. We're going to find out how. So again, DNA is so small we can't even see them under a microscope. Imagine if we had a bunch of different shoelaces of different lengths. That would be really, really easy to sort out. You just stretch them out. All right, this one's bigger, this one's smaller. But DNA is tiny, it's on a nanoscale. So gel electrophoresis is a way that we can sort those out. And it's a very simple process. It's not, I don't know what you would think of in order to do that, but it's not something I would think like, oh, here's an interesting way to do it. But it really is a great technique. So we'll sort them out by length, as I said before. Now let's find out how it works. So initially we're going to start with a gel. It's all like Jello, kind of like a sponge made of Jello. Got all kinds of little tiny little holes in it, as you can see with the different textures here. And it's got these wells up at the top. So let's see what we're going to do with this. So in each of these wells, we can actually put our DNA. As you can see, DNA, one end of the gel. So one end of the gel has the wells with the DNA. There's a negative charge on this end because, as you may or may not know, DNA actually has a negative charge. So two negatives together are going to cause the DNA to move away towards the positive end of the gel. 1 second. Hey, cow, dogs. Sorry, guys. Dogs are acting up. It happens. Alrighty. Now, as the strands begin to move through this Jello sponge filled with holes, the shorter ones are going to have an easier time moving through. Just imagine if you were running through an obstacle course with all kinds of crazy obstacles everywhere. The smaller, faster, and lighter you are, the more easily you're going to be able to make your way through the obstacle course. Like, you guys could kick my butt in the obstacle course. It's going to take me a while. I'm a big dude. It takes a lot of energy to move. I got to move through all this stuff. So you guys will be able to reach the end before me. And pretty much that's just kind of how it goes with the DNA. The smaller strands move much more quickly through the gel than the larger ones. So at the end of it, when we actually stain and look at it, we'll see clumps towards the top and as they'll trickle down to the bottom. And then we know, okay, the further they've moved, the smaller they are. The less they've moved, the bigger they are. Very simple concept here. So we're actually going to see it visualized right there once we dye the gel so we can see it with our naked eyes, and you can see all kinds of ones. So these we know are going to be very small. These ones at the top, much larger. These ones in the middle are, yeah, right about there in the middle. All right, so now we're going to do it ourselves. We're going to be real scientists now, and we're going to do what's called running a gel. So if you ever hear a scientist say they need to run a gel, this is what they're talking about, gel electrophoresis. So first we have to make the gel. We have to set up the apparatus. We have to load our samples in, hook up the electrical currents and run it. Then we're going to stain it and analyze the results. Are you ready? Are you ready? Are you excited? Me too. Okay, let's make our gel, guys. Now, we're going to need a few different things to make our gel. It's a lot like making Jello. I wouldn't personally eat this gel, but, you know, to each their own. So we're going to need some powdered agarose. That's right there. It's like gelatin, but it's made from seaweed. We have our buffer. It's going to be a salt buffer. Helps the electricity to move through more quickly. We have a flask. That's where we're going to mix everything in. We got a microwave. Just your average microwave, but again, I wouldn't cook food in it or burritos because, you know, come on, you're using it for lab stuff. We also have our gel mold and our gel cone because, you know, gel needs to look good. God, a terrible Monday joke. All right, so let's go ahead and make our mold. We're going to take some of the seaweed agarose, like I said, a lot like gelatin, but made from seaweed, so it's vegan. And then we're going to take our saltwater buffer. We're going to pour some of this guy in here. Again, this saltwater buffer is going to allow the electrical charges to flow through the gel even better. I'm going to pour that in, shake it all around, shaky, shaky, mixy, mixy. I'm going to put a little bit of plastic on the top just so the gel doesn't boil over because that makes a big mess. I'm going to open up our microwave, pop this little dude inside, and let's get her going. We want it nice and hot so everything mixes in. Again, just like you were doing Jell-O, usually you would do that on the stove top. This we're going to do in a microwave. I guess you can do Jell-O in a microwave too. All right, it's all mixed. We're going to be very careful with our hands. We don't want to burn ourselves using protective equipment, PPE. All right, we just poured that in there. Now we need our gel comb, make our gel look all pretty. This is actually going to make the wells. So we're going to pop this guy in there. It's going to make those tiny little well holes in the top. We're going to let this whole thing cool, and then it will harden into a gel, and then we'll carefully extract that comb. All right, so it's cooled. We're going to take our comb very carefully. We don't want to break the gel. All right, looking good, son. Okay, now we have our electrophoresis box. We're going to need some more of that buffer because, again, we're going to be running a current through the liquid and through the gel. So the saltwater buffer is going to help that current move much more effectively. Let's see, gel. Oh, first we need to set up our box. So we're going to need the gel and the thing of buffer. Oh, it doesn't want me to touch it yet. Fine, I won't touch it yet. All right, so we're going to pour our buffer into our electrophoresis box. That's our saltwater buffer so that the electrical current can move through more efficiently. And we're going to place this whole apparatus inside. Whole thing's going in there. All right, now we got a party, guys. All right, now we need to load our sample into the gel. So we're going to need some loading buffer. We're going to need the tube of DNA, a size standard, and a micropipetter. Now I'm going to describe the size standard in just a little bit, okay? So let's go ahead and we need to get our pipette tip because we need to keep everything sterile. So we don't want to take our pipette and dip it into a whole bunch of different stuff because it's going to contaminate it. So we get a fresh pipette tip. We're going to add it to the sample. Now the samples are prepared in a clear liquid solution, and it would be hard to see if we try to load it directly in. So the loading buffer has a dye that makes the sample easy to see. It's also a little bit goopy, which makes the sample thicker, so it'll drop into the well instead of floating away. So again, these are all kinds of little things that have been thought of by scientists and great minds. So we're going to take a loading buffer, we're going to add it to our DNA, and now we're going to use the pipette to transfer the sample into the well. So we need to suck some of this guy up. And remember, it's got a little bit of that blue dye. It's got some gel in it too to make it nice and heavy. Now in real life, this is kind of a difficult process because you have to, these tiny little wells, you have to put this just far enough to get it in, but you don't want to press too hard or it's going to disrupt your gel. So just like that, look at how well we did. It takes some practice in real life, and we would be doing that if we were in class, but you guys will have definitely a chance to do that in high school and stuff like that. It's frustrating at first a little because it does take some practice, but it's actually kind of fun once you start to get it. All right, so now we need our DNA size standard. And I'm going to pick some up with my pipette. Now, the whole point of the size standard is so that we can compare our DNA to something that we know the sizes of. So let's go ahead and do that same thing. Careful, careful, careful. Load it in. So we already know what sizes the DNA standard is. So when we actually dye the gel and we can see it, we'll already know what sizes the DNA in the standard are because it's standard. That's what it is. Then we can compare those sizes to our DNA to get an estimate of how big it is. Now, again, we are going to need electricity to run through the gel. The red represents positive, the black represents negative. DNA is negatively charged, so if we put a negative on a negative, it's going to repel it, and it's going to be attracted to the positive side. So let's make sure our black cord is on the side closest to the DNA. So we're going to hook that guy up, hook our positive up, and let's run this dude. Hey-oh. We're going to see some bubbles. That just means it's working. So we're good, guys. Currently, everything is running. Everything is looking as it should. Well done. Again, repelled by the negative charge, DNA moves through the gel towards the positive charge at the other end. The short strands move more quickly than the long ones do. Over time, the shorter strands will migrate further from the starting point than the longer strands. Now, we can't actually see them migrating, but when we dye the gel at the end, we will see the path that they took. Let's go ahead and do that, shall we? So we finished running our gel. We've taken the mold out. Time has come to analyze our sample and estimate the lengths of the DNA strands. We're going to use an ethidium bromide solution. This is a staining chemical, binds to DNA, and it shows up under a fluorescent light. So even though we can't see single strands, we will see the large groups of stained DNA, and these will show up as bands in the gel. So we're going to go ahead and take our gel out very carefully, and we're going to put it in this staining solution, and leave it in there for about 30 minutes. So why don't you guys chill, and we'll just sit here and wait for 30 minutes while it does it. Hopefully nobody ran away, because seriously, that'd be ridiculous. All right, so we're all stained and good to go. Now we need to use an ultra UV fluorescent type of light. Pop that dude on the box, click it on, and what are we going to see? Hey, we got strands. Now see, here's our DNA standard. We know how long these are. We know this is 1,000, 2003. How do we know that? Because it was manufactured to be these exact sizes. So when we look at ours, we can compare it to see about how large it is. So in this case, this band is very close to this other band here. About 6,000 base pairs. Okay. Let's go down to this one. This is kind of between it, maybe a little closer to 3,000 and 4,000. So let's go with, I don't know, what do you think, 3,450? Yeah, me too. Down here, it looks to be right about in the middle, between 2,000 and 1,000, so let's call that guy 1,500. Now let's see if we're right. Fingers crossed, guys. Oh, estimates are correct! Estimated length of each band in your DNA sample is based on how they line up with the bands of the known standard, right there. Now, these are just approximations, but again, even these approximations will give us a very good idea of what we are dealing with. So congratulations, guys. Y'all have just run, you made your gel, you prepared your DNA, you put them in the wells, you ran the gel, and you analyzed it. Congratulations, guys. And I actually mean that sincerely. Anyone who is sitting right now, watching these videos, trying to learn something, and to just make themselves better, you've already won, guys. Congratulations to you. Boom, shaka, laka. You have run a gel. Now, what you guys should do is go to the PCR lab that we did last week just to refresh your memory a little bit. Make sure you run through all these yourselves. There will be a guided worksheet to go along with it. This will be a graded assignment, so please, you can do it as you're going through it. Maybe you want to go through it once by yourself and then go through it. Maybe you want to go through it once, then use my thing again. Whatever you guys want to do. Make sure you fill out the guided worksheet, make sure you review everything, and be ready for a quiz later on this week. I will most likely put the quiz out on Wednesday, and it will be due, like everything else, by Saturday evening. All right, everybody, thank you so much for spending some time with me. Make sure y'all check out the PCR virtual lab as well to refresh your memories. Stay safe, everybody.